So this is the second part of preventing and controlling virus diseases. We're going to talk about antivirals. And as you know from last time, vaccines have been very successful at preventing disease. But if you are already infected, they have modest effect. Although we're trying to develop a vaccine for AIDS that may be used for people who are already infected. But in general, if you're already infected, a vaccine is not going to do much good. So what we do in that case is to try and give you an antiviral drug that, in theory, can stop an infection that's already started. And there are a number of situations where that might be the case. The most obvious, of course, is where there's no vaccine available for a particular infection, but other situations arise as well. Antiviral research has been going on for about 50 years, and even despite that, there are very, very few antiviral drugs available. There are about 30 or so on the market today, and most of them are against HIV. The large part are against HIV and herpes viruses, and a few assorted other viruses. We're going to talk a little bit about why this is so. And this table shows some selected, available, approved compounds organized according to the target in the replication cycle that they inhibit. So by now, in this course, you should understand every step of the replication cycle. So you should be able to understand what we mean by all of these terms: uncoding, DNA polymerase, reverse transcriptase, protease, neuraminidase, IMP dehydrogenase. So for example, we have uncoding inhibitors for influenza viruses. We also have others that aren't shown on here that attack earlier steps, attachment. We have inhibitors of HIV. We'll talk about those later. There are a number of inhibitors that target the DNA polymerase of viruses, or the reverse transcriptase, as the case for HIV. And we'll talk about the mechanism of action of some of those. These are mainly base analogs, compounds that mimic a base, but there are other mechanisms as well. There are some inhibitors of viral proteases, and there will be more coming on the market soon. Neuraminidase inhibitors, which you probably heard a lot about in the last year for influenza virus. And then ribavirin, which is a very broadly acting inhibitor, apparently inhibits a number of viruses and currently used mainly for hepatitis C and respiratory syncytial virus infections. So why are there so few antiviral drugs available? Well, the problem has to do with the fact that viruses are obligate parasites. They use a lot of what the cell provides. So any compound that you make that interferes with the ability of a virus to grow has to be specific. If it interferes with a host protein or a host enzyme of some sort, it's going to have side effects. And that's not acceptable for any pharmaceutical. So that's why one of the main reasons we don't have a lot of antiviral drugs. Every step in the cycle engages a host function. Therefore, it's very difficult to make an antiviral that's specific for a viral protein. The other reason is that a lot of viruses are quite dangerous to work with, and so screening antivirals has been up until now difficult, although that's changing. Some of them can't even be grown in culture. The viruses cannot be grown. So for example, hepatitis B virus to this day still can't be grown in culture as an infectious virus. Hepatitis C only recently is able to be grown, as well as human papillomaviruses. But there are ways to get around this, as you see. You can identify inhibitors, although in the end it's nice always to have a culture system with an infectious virus that allows you to test them. Sometimes we don't have animal models for testing disease, and that makes it difficult, because one of the things you'd like to do when you have candidates is to see if they work in an animal model. So that's a problem. And then if you have very dangerous viruses like Ebola and Lassa and even smallpox, very difficult to work with, even under high containment. Accidents always happen, so you have to find other ways around it. So these are some of the reasons why we don't have a lot of drugs. But the third reason is probably the most important. The drug has to be almost 100% effective at blocking virus replication. There's no room for error here. It has to be potent and effective. Now, many of the other pharmaceuticals that you can buy, prescription drugs for a variety of ailments, non-infectious ailments, for asthma, for example, these don't have to work 100% to give you therapeutic benefit. Okay, so sometimes the enzyme activity, which is the target of these drugs, is only partially blocked, but you have a very good therapeutic effect. But that doesn't work with viruses. You can't have partial inhibition of replication. And so it's very difficult to do that, and that's another reason why we don't have a lot of antivirals. So the drugs must be potent and safe, and that combination is very difficult to achieve. And as a consequence, finding drugs that fit both criteria, it takes a lot of time, can take up to 10 years to get a drug to market, and it's very expensive. That's why in the end they cost a lot of money. So if you don't block replication completely, you'll get resistance, as we will see. So if you have an inhibitor that's not 100% effective, it allows viruses to multiply, that's a perfect way to get virus resistance, because as the viruses multiply, they make the mistakes that lead to resistance, and will always happen. And this is a graph that illustrates this. This is a graph of the amount of virus present with time after drug treatment. And there are 3 different doses of drug given, a low, an intermediate dose, and an optimal dose. So the optimal dose of drug blocks virus replication completely, and so the load goes down to near 0. The virus can't replicate, and if it can't replicate, it can't make mutants that might be resistant to the drug. So that's the optimal situation. Intermediate dose, you get initial inhibition of replication, but it's not enough to completely inhibit all the viruses, and you get emergence of resistance. And the same for a low dose. A slight inhibition of replication, and then the mutants in the population that are resistant to the drug predominate. So you always have to have the optimal dose for the drug, which is one of the things that is tested from the very beginning in cells, in animals, and in humans to find out what's the best dose for inhibition. Another problem is, if those 2 weren't enough, is that many acute infections are short by definition, as we talked about before. So it's very difficult to intervene with a drug. By the time you feel sick, the virus is peaked or is beginning to go down in its titers in you, because, as you know, the symptoms are immune response mediated, not correlating with the amount of virus. So you feel sick, and in a few days you get a prescription. It's too late for the drug to be of any use. Remember, the antivirals against influenza you have to take within 2 days of your first symptoms, otherwise they're useless to take. So you have to give these antivirals for these acute infections early in infection, or you have to give them to everybody, which is prophylaxic use, and that's not really a well-endorsed usage. Giving drugs to healthy people is generally a bad thing, and so we don't do that typically. There's always a lot of discussion about it. We don't have any broad-spectrum antivirals yet approved. There might be some coming down, but so far we don't. So every virus infection has to be treated specifically. It would be great if we had one antiviral that inhibited all infections. Then if you got sick, you just take it, and you don't even worry about what virus it is. And that's what they do with antibiotics. You often will get sick, you get a prescription for an antibiotic. It may not even be a bacterial infection that you have, but you get an antibiotic anyway. It does nothing for you, and it just increases antibiotic resistance in the population. But we don't have any of these yet available. So in terms of these acute infections, continuing on this problem area, we don't have the ability to identify the viruses causing most acute infections anyway. Influenza is an exception, as we've talked about, but the diagnostic kits that are quick are not very good. And for the other virus infections, we don't have any diagnosis. But if we did, if you could diagnose something, it would still take time to get the identity, get a prescription filled, and all of that, and most people simply wouldn't be taking the drug in time to have an effect on the infection. So this means that you can't prescribe a drug because you don't know what the infection is, or if you can, it's too late to do any use. And again, getting back to this point that I talked about before on the acute infections lecture, the absence of diagnostic reagents is a big limitation. If we had quick diagnostic kits that were accurate, there would be a much bigger arsenal of antiviral drugs available. And until this happens, and it will happen someday, there aren't going to be many. No point in making antivirals against rhinoviruses if you can't diagnose them. The cold is over in a week. There's just no way an antiviral is going to be useful. So let's talk a little bit about the history of antiviral research. It first began in modest ways in the 1950s. So by this time, as you know, there were antibiotics that were used to treat bacterial infections. Those had begun in the 1930s. And some of the first ones were the sulfonamide antibiotics. So chemists started to modify those and see if they would inhibit viruses. Thiosemicarbazones, a certain class of chemicals, were looked at for antiviral activity, and some were identified in the 50s that were active against pox viruses. Now, smallpox was at the time a major issue. After World War II, still infecting many, many people globally, hadn't been eradicated. There was always the possibility that it would be brought into places where immunization was controlling it. So, for example, in 1967, as recently as then, millions of people were still dying of smallpox. So there was impetus to make antivirals against this virus. In the 60s and 70s began the so-called blind screening programs to find chemicals with antiviral activity. This was really, again, the impetus was from the success in antibiotics for treating bacterial infection. So what is blind screening? Exactly what it sounds like. You just look at big collections of chemicals and try and find ones that inhibit virus replication in cell culture. There's no attempt to even focus on a virus or a specific mechanism or step in replication. And typically these were collections of chemicals that companies had stored away, or even natural product mixtures. You go out into the field and you collect moss or dirt, and you bring it back to the lab and you make extracts, and you test them for antiviral activity. And you test a whole lot of viruses, not just one. That's why it's called blind screening. So this could give you a hit. It could give you a compound or a mixture that blocks replication in cell culture. Then you start to fractionate it because you'd like to identify what is in the mixture that is inhibiting replication. You can go on and test it in animals. And then you identify molecules, which we call leads. And then you begin to modify them. The chemists then take them and modify them in a variety of ways to try and increase their potency, reduce toxicity, make them more bioavailable, and improve their general properties. And this is a very common approach to making an antiviral. You get a lead that is never the ideal compound, but then when you modify it in the end, you may get something that works well. In these kinds of blind screens, you go through thousands and thousands of molecules, and even before you ever get into people. You go from thousands and thousands to maybe one molecule, which you then modify, and this can take years and years. And this is not at all automated. As you will see now, it can be automated, but back then it was all done by hand. So as you can imagine, there was a lot of work involved here, but not a lot of success. One of the exceptions was this drug called Simitrel, or amantadine, which was approved in the 60s for treating influenza A virus infection. So this was picked up in a blind screen, turned out to be very effective, and it's still being used against influenza A viruses. But we didn't know the mechanism of action back in the 60s. It was only learned in the 90s, and we'll talk about that a bit later. So today we have very different approaches to discovering antivirals. We have recombinant DNA technology, which allows us to express individual viral proteins. We can look for inhibitors. We have very sophisticated chemistry so that we can make hundreds and thousands of different compounds very quickly. It's really matured quite a bit. So you can clone viral genes and express them in organisms, purify them. We can get structures to help predict what kind of drugs we should make. So we don't do blind screening anymore. This is just too diffuse. We pick a virus, and not only that, we pick a specific protein in a virus and start looking for inhibitors. And we can do that because now we know the life cycles of most of the viruses that we want to intervene. All the information we've been talking about in this course, all the steps of the life cycle, that's known in great detail. So we can pick targets and design how to inhibit them. And even for viruses that you can't grow, you can find inhibitors. You can pick a protein, an enzyme of the virus, for example, and express it in a cell culture and look for inhibitors of it. And if you don't have the ability to grow the virus, you could then proceed into safety testing and eventually into people, although it would be nice for those viruses to be able to test them in cell culture. So these are some of the steps that are targets for antiviral discovery. This is your typical replicative cycle. What virus is this? Anybody know? What family? Sorry? All right, let's see here. Is it plus or negative stranded? Let's narrow that down. It's plus, right? Because it's being translated right away. And what other clues? It's a icosahedral capsid, a plus-stranded icosahedral capsid. So there are a lot of choices there. Polio. It's polio. I was going to say you could just take my biases and guess, and that would be it. Yeah, it's polio. This is the most beautiful and simplest replication scheme. So you have attachment inhibitors. You have inhibitors of the inhibitors based on interferon, which can inhibit at a variety of steps. You can have inhibitors of the genome replication cycles, which we'll talk about. And you can have inhibitors of processing, the so-called peptidomimetics, which inhibit HIV, and even inhibitors of release, the neuraminidase inhibitors of influenza. So you pick your target, you decide what is the most likely to give you an inhibitor. Maybe you have the most information, structural information, and you go after that. And this is a sort of outline of the path of drug discovery, just to give you an idea of what is done in a company to make a drug. You identify a medical need, you find a virus that causes a disease and for which you need an antiviral compound, and then of course you need to do some research to identify mechanisms that you want to inhibit. You pick out what part of the replication cycle you'd like to focus on, and then at some point you have to do a proof of principle. Let's say you pick a viral protease that you want to inhibit. A proof of principle would be showing that removing the protease prevents the virus from replicating. Right? If you remove the gene and there's no effect on virus replication in cell culture, then you shouldn't make an antiviral directed against that. So that's what we mean by a proof of principle. So then you identify a mechanism. Maybe you have some structure data that will help guide you. Or you can devise mechanism-based screens to look for inhibitors. You have libraries of compounds which you put through these screens and you get hits which are developed into lead compounds. And these are modified by the chemists, and you put them in your assay over and over again. And the most promising ones will go into animals to test for toxicity. If you have an animal model for your disease, you can test efficacy. And eventually a drug candidate will go into people. And there are many, many parameters that have to be right, not just the ability of the compound to inhibit in cell culture. So this whole process, this discovery process, doesn't find drugs. A drug is what we define as something approved and licensed in humans. All the discovery process does is detect hits. And the hits are then developed into leads, which eventually are developed into a drug. So these methods that we're going to talk about today in a little bit of detail are quite a long way away from drugs. All they tell you is that in a cell culture or in some kind of assay, which may not be during a replicative cycle, that the drug can inhibit. These kinds of questions you have to address. You have to say, is it bioavailable? If you take this drug orally, is it going to go where you need it to inhibit that particular virus? So if it's a respiratory infection, will it get to the respiratory tract if you take it orally? You can't assume that it will. You have to test that. What are the pharmacokinetics? Does it have a long half-life? Does it last a long enough time to be effective with respect to the growth curve of the virus? And will it be safe? Is it toxic? If it has side effects, you throw it away immediately. If it kills your cells in culture, that's it. Forget it. You throw it away. So a lot of these questions are all addressed during the development process. And so that's the difference between research and development. Developing a lead compounds means you do some research to find a lead, and then you develop it into a product. And this can cost $100 to $200 million and take up to 10 years. You start with thousands of leads, and you break them down until you get one or two. And at the end, the kinds of studies you have to do are very expensive. So you can't do it. You can't do these studies in animals and humans on dozens of compounds. You have to pick one or two. You may have lots of leads that look good, but you have to make the decision to pick one or two that you think will go forward. Not an easy decision. So these are the steps in taking an antiviral lead through and forward to development. You have to do safety testing, and most of the compounds fail right here, either in cell culture and in a small animal. They're too toxic and you throw them away. If it passes safety, you have to be able to make a lot of it. So the chemists have to get involved and say, yeah, this could be easy to make, or no, forget it. How the drug is formulated, an injection, an oral formulation, what kind of capsule, how the drug lasts, as we said. And finally, the last test, if you're lucky, you go into clinical trials in people. And clinical trials generally occur in at least 3 phases of increasing complexity and cost. So 5 to 10 years after your initial lead, which you had discovered in the screening process. So here's another way to look at this, all these hurdles in the way of finding drugs. So here, this is the number of compounds on the y-axis. You could start with libraries of 100,000 compounds very easily. And then you just put those into a cell, either an infection model or an enzyme inhibition model, and look for some kind of inhibition. And you reject some right away because they don't do anything. Then you look at toxicity, and you throw away the ones that kill your cells. You go to animal models if you have them and ask, does it have an antiviral effect? You take the ones that do, and you throw away the ones that are toxic in an animal. So they weren't toxic in cells, but they are toxic in an animal. So now you have a lot of animal data which maybe justifies going into people. You look for safety in people. You look for an antiviral effect. And eventually you get down to one compound that you may have approved for general use. So a lot of money and a lot of time, but it may not be approved. It may be rejected by the regulatory agencies. So it's not easy work. It's another reason why we don't have very many antiviral compounds. So let's look at a couple of examples of screens that you could develop. There's so many clever ones that have been put together to screen viral proteins for inhibitors. And this one is a mechanism-based screen where we're looking for a protease inhibitor. So we have a viral protease that we know is essential for replication, and we want to find a small molecule that inhibits its activity. So what has been done here is to make a small substrate for the protease. So we have to know the protease cleavage site. And this substrate is unusual. It has a bead at one side, a big bead, so that we can separate it physically by centrifugation. And it has a little green light at the other side. It has a fluorescent compound that will glow under certain conditions. So when the protease works, it cleaves away the green light from the rest of the molecule, and you can separate it very readily by spinning out the insoluble part, and you can measure the fluorescence intensity of this thing being released in the soluble compartment. So you take out the insoluble stuff, the uncleaved stuff, and the cleaved material, and you see the activity. And then you can use this to screen for inhibitors. It's a very nice assay. It's colorimetric, and it's very quick. So that's one example. That would be done in a 96 well or a 1500 well plate. It's an in vitro assay. Here's an example of an assay that you could do in cells. This could be done in bacteria. And here we have bacteria that are tetracycline resistant because they have a protein in the membrane that pumps out tetracycline, so the bacteria can grow in the presence of it. And what has been done here is to engineer into this tetracycline pump a cleavage site for the HIV protease. So when you express HIV protease in these cells, it renders them tetracycline sensitive. Just by cleaving this one loop of this transporter molecule, it makes them tetracycline sensitive. So then you just incubate these bacteria with different compounds, and you look for bacteria that are resistant to tetracycline. And that would be those that are where the protease is inactivated. So that's a cell-based screen as opposed to one done purely in vitro that we talked about before. Now this one I just found and added this morning because I want to show you how to look for antivirals using viruses that are really dangerous. So there are some paramyxoviruses which originated in Australia, down that part of the world, which came from bats. And these are the Hendra and Nipah viruses, which cause fatal encephalitis in people. So these are very dangerous to work with. But in certain conditions, we'd like to have an antiviral to protect certain at-risk populations. So what you do is you make a pseudotyped virus. You take the genes encoding the glycoprotein of these dangerous viruses only, and you express those on another virus particle. So you could use vesicular stomatitis virus, for example, expressing the Hendra virus glycoprotein. So now it's not a dangerous infection to work with. You can use this in a laboratory and screen for inhibitors. So you take this pseudotype virus, you can screen for inhibitors of fusion into the cell, for example. And there are two different ways of using this, which are very clever here. You can take these pseudotype viruses. Remember, the genome is VSV. There's no Hendra virus genome here. The glycoproteins came from the cell where this virus was made. This is called a pseudotype virus. You can infect cells that are permissive for Hendra, and you can assay entry and look for inhibitors of that process. But these viruses, when they go into cells, they'll replicate, and the replication is largely VSV, so you don't want to have inhibitors of VSV. You're looking for inhibitors of Hendra. But what can be done is you can express Hendra virus glycoproteins on this target cell. You infect with your pseudotyped virus, and then this virus, which is VSV, will get in and start replicating and producing particles, and these particles will have Hendra virus glycoproteins on them. So you can test for the production of inhibitors of the production of Hendra virus as well. So you can test for entry inhibitors and release inhibitors. So this is what you do for a virus that's very dangerous. You take the glycoproteins and you put them in a pseudotype virus and develop screens for inhibitors. Now we do high throughput screening. I don't do high throughput screening. I do low throughput screening. But in companies, they buy very expensive robots where you can do very, very high automated screening. So this is a 1,500 well plate. These are all tiny wells, and it's on a conveyor, and there's a robot that will pipette into it automatically all the reagents that you need to carry out the assay. And then there are these robot arms which will pick up the plates individually and put them in incubators, so no one even has to be present here. This can run 24/7. Material is put into the wells, the robots pick them up, put them in an incubator for X time, they take them out, read the experiment, you come in the morning and you get the results. So it's not dangerous and it's high throughput screening. But these are like the kinds of robots that put cars together. When they're finished putting cars together, they go to the pharmaceutical companies and look for antiviral drugs. Wrong one. Okay. So antiviral screening today, high throughput, you can do 10,000 or more compounds a day. We use libraries, chemical libraries, which are synthesized, and there are ways to make this very diverse collections of chemicals. We're not going to get into that. You can still look at natural products. You can go out in the environment and look for things, but people do this less and less. Combinatorial chemistry is a way of taking the components of molecules, the Legos, and putting them together in a very interesting way to get thousands and thousands of variations. We can use structure-based design, which means you solve the X-ray structure of a viral protein, and you look at where the natural ligand fits, and you design inhibitors that would fit in there. We'll look at an example of that. And then you can do in silico screening, where you have a computer just take the target in a whole library of compounds and see which fits, and then start the screening that way and narrow it down instead of going through thousands and thousands of compounds. So very sophisticated screening. But what's not sophisticated, of course, is that we get resistance. No matter what technology we have to get drugs, we will always have resistance. I would say it would be a good bet to say yes to the question, will antiviral resistance occur? Okay, so you always have to anticipate it. So you need to have a lot of antivirals around to get around that. And why is that? Why do we get resistance? Because, as you know, viruses replicate really well, and they have really high mutation frequencies, which we will talk about more next time. And this is a big problem in chronic infections that go on for long periods. HIV, hepatitis B, hepatitis C. Not the acute infections that are over in a short time as much as these long-term ones. Every antiviral we have today that's licensed, there are resistant mutants. And everything that we're ever going to make is, in fact, there will be resistant mutants as well. So this today is a problem because we don't have a big arsenal except for HIV. But in the future, if we have more antivirals, we might be able to get around this. So why is drug resistance a problem? We've been talking about it a lot. But what's the practical consequence? Well, if a patient displays resistance to a particular antiviral, you can't treat them any longer with that. It does no good to treat them. Yet the infection is continuing. And of course, if you don't have anything else, then that's the end of the story. You can't treat the infection. It's not stoppable. There's a lot of work understanding the basis of resistance. And the idea is that if we can understand how resistance evolves, we might be able to design antivirals that get around it. So you will see lots of papers in the literature trying to understand the basis of resistance. Maybe we can devise new strategies. So how does drug resistance occur? Well, RNA viruses, as you've heard many times, have an RNA polymerase that makes mistakes. Actually, all nucleic acid polymerases make mistakes, but the RNA polymerases don't have correction mechanisms. So they incorporate many mistakes into the nucleic acid product. And typically, for RNA viruses, you get one misincorporation in about every 10,000 or 100,000 nucleotides that are polymerized, right? And that's quite a bit more than the error rate of the host DNA genome. Now, if that means that in an RNA genome of 10 kb, which is about average, you get one mutation in somewhere between 1 to 10 genomes. So if it's 10 to the 4th, you can have a mutation in every genome that is produced. So when you make 10,000 genomes, that collection would have a mutation at every position, essentially. And so there would be every 10,000 viruses would have a resistance to a particular drug, in theory. DNA viruses also make mistakes. The polymerases make mistakes, but they have accessory proteins that can replace the misincorporated nucleotide. So they evolve more slowly because they have less diversity. So here's an example of this error correction process. Here's a DNA template, and it's being copied by a DNA polymerase, and the product is being made in a 5 to 3' direction. Here there's a misincorporation. The polymerase put the wrong base in. It happens. Everybody makes mistakes. But DNA polymerases have erasers on their pencils. They have a 3 to 5' exonuclease, which will chew back this area until the mismatch is fixed, and then the polymerase will go back and fill it in. And this happens all the time so that you have fewer mistakes. But the RNA viruses, the RNA polymerases can't do this. They can't correct their errors, as far as we know. So let's talk about resistance in terms of a specific drug. Let's pick acyclovir, an anti-herpes simplex drug, which you will get if you have herpes and you go to get a prescription. You'll get acyclovir or one of its derivatives. And we call acyclovir a prodrug, which you'll see the basis for that in a moment. And it's a nucleoside analog. So the nucleic acids are constructed using one of the 4 bases, which are shown here, A, G, T, and C, or U if this were RNA. And analogs of each have been made, and they're all shown here, which are antiviral compounds. And the analogs are incorporated into the growing nucleotide strand, but they are not extendable, as we'll see. So here, let's take a look at acyclovir, which is a derivative of guanosine. You can see how is acyclovir different? Well, it has the same base structure here, but it's missing a good part of the sugar, of the ribose, and this hydroxyl down here. The hydroxyl, the 3' hydroxyl, of course, is where the next base would be added in a growing chain. So once acyclovir is incorporated into DNA, synthesis will stop. That's the basis of it working. And a modification of acyclovir, which was subsequently made, is called gancyclovir, and you can see it has an hydroxyl here. And these have other properties that make them preferable. Okay, so many antivirals are nucleoside or nucleotide analogs, such as shown here. So acyclovir works in the following way. In order for nucleotides to be incorporated into DNA, they have to be phosphorylated. So acyclovir can be taken up into the cell. Phosphorylated compounds in general don't get into cells very well, but this is not phosphorylated. That's why we call it a prodrug. It's taken up into the cell, but it's not recognized by any cellular kinase unless the cell happens to be infected with herpes simplex virus or a virus that makes a thymidine kinase, a TK, in which case the acyclovir will be phosphorylated. So you see, this makes it specific for virus-infected cells. Uninfected cells, the acyclovir will get in, and it won't be phosphorylated, so it can't inhibit DNA synthesis. Once the drug has a single phosphate, then two other cellular kinases add the second and the third phosphate, and then it's ready for incorporation into growing DNA by the viral polymerase. And again, because it's missing the 3' hydroxyl, once it's incorporated in the growing chain, the synthesis stops. So it's a chain terminator. It inhibits DNA synthesis. It does so in virus-infected cells, and that's how virus replication is inhibited. Now, you get spontaneously during virus replication resistance to this drug. Yes?
I'm sorry, just go back to the mechanism. You're saying that when it's taken up into cells, it's not recognized if you're not infected?
If it's taken up into all cells, if it's not infected with herpes simplex, it will not be phosphorylated, so it will not proceed along this line of inhibition. But in a virus-infected cell, it will be phosphorylated and incorporated into either viral or DNA, or cellular DNA, but it's a virus-infected cell, so you don't mind if you inhibit cellular polymerase. You get resistance arising during virus replication. There are two kinds of mutants that you get. You get some mutants in the viral TK gene that do not phosphorylate the prodrug, so they can't recognize it and phosphorylate it, so it doesn't get taken up into growing DNA. So that becomes resistant. Or you have mutations in the viral DNA polymerase gene, which block it from incorporating the acyclovir triphosphate. So in these cases, the TK is not mutated, the viral polymerase is mutated, but doesn't incorporate the phosphorylated drug, and therefore it's not inhibitory. So two different classes of viral mutants that arise. Now, in a lot of people, these are not a big deal because these mutants tend to be a little sicker than wild-type virus, unless you have AIDS or some other immunosuppression, in which case these TK mutants can be a big problem because they can't be treated. And they can cause disseminated disease, and they're often resistant to other analogs that you could otherwise use to treat infection. There is a second possibility to use in individuals with TK mutants, that is to treat with a DNA polymerase inhibitor called foscarnet. It's not as good a drug as AZT, as acyclovir, because it has side effects. But if you don't have any other choice, you have to use it. But sometimes if the acyclovir resistance occurs by mutations in the polymerase gene, they will also be resistant to foscarnet, in which case there's nothing else that you can do. No treatment options left. So that's how it works, and that's how you get resistance. Now here's an example of how you would modify a drug to make it better. Acyclovir is a very good drug, but it's not so bioavailable if you take it orally. It doesn't absorb well from the stomach and intestine. But chemists figured out that if you add an amino acid here, valine, right here, it's more bioavailable. And you can take it orally, and it's absorbed very well. In many cases, acyclovir or other derivatives are used topically. But if you want to use it orally, then you use this drug, valacyclovir. And this was just determined empirically by modifying acyclovir and testing it in animals to be more bioavailable. So this is actually a pro-drug, because when you take this, first the valine is cleaved off to make acyclovir, and then it has to be phosphorylated to become the active drug. The other drug we'll look at in some detail is an inhibitor of influenza virus, amantadine or Symetrel, which was an early discovery, as we talked about. This drug interacts with the M2 protein of influenza virus, which forms the ion channel in the virus itself, in the virus particle. So you remember, influenza virus is enveloped. It has two glycoproteins in the envelope, but it also has, at very low numbers, a channel. And you can see one of them in this particle right here. That's the M2 ion channel. And that works during uptake of the particle. The virus binds a receptor. It's taken into the cell by endocytosis. And then the endosome begins to acidify. Eventually, the acidification is going to lead to fusion of the virus in the endosome membrane. But while the acidification is going on, the pump, the channel in the particle is also working. So how does the pH in the endosome drop? Well, there's a pump in the endosome that pumps protons into the interior that lowers the pH. And then those protons are then further pumped into the interior of the virion by this M2 ion channel. And that is essential for uncoating. It's not essential for fusion. The fusion can happen without the M2 protein. But it's essential for releasing these RNPs out of the membrane, and so they can diffuse into the, or be taken into the nucleus to do RNA synthesis. So if you inhibit this ion channel in the virus, when fusion occurs, all these RNPs remain stuck to the membrane. The M proteins remain here, and the RNAs remain stuck on them, and they never get into the nucleus. So that's how it inhibits virus replication, by blocking the ability of the viral RNA to get into the nucleus. Okay? So that's the M2. So amantadine inhibits this by blocking the channel. But when this drug was discovered, the mechanism wasn't known. This was figured out many years later. So this is a close-up view of the channel in the virion membrane. So this is the virus membrane. This is the M2 protein. It's quite a short protein, and 4 of them form a channel. Only 2 of them are shown here. And through this, the protons are pumped. So this is an active channel that puts the protons through it. Amantadine is shown here schematically. It binds the M2 protein and plugs up the channel so the protons cannot get through. That's a very simple mechanism. It physically blocks the channel. Now, this drug interacts with very specific amino acids in this channel. So how do you think you get resistance to amantadine?
You just get a mutation in one of those proteins that doesn't affect the function, doesn't allow amantadine to function.
Yeah, any of these amino acids that interact with amantadine, you change it, and now the amantadine doesn't attach anymore, doesn't block the channel, resistance. And in fact, those are what is seen. And now the H3N2 viruses, which circulated up until last year, they were 100% resistant to amantadine by virtue of these changes in the M2 ion channel. We have another class of inhibitors of flu, the neuraminidase inhibitors. You remember neuraminidase, which is one of the glycoproteins on the virus. Its main function is to help release budding virions from the surface. So as new virions form, remember these have a hemagglutinin in them which would bind to sialic acid on the surface. So the newly made viruses would just stay on the surface if it weren't for neuraminidase, which removes all the sialic acid so the viruses can be released. Okay, so the neuraminidase is important for the release of the virus from the cell surface. The inhibitors block the neuraminidase function, so in the presence of them, the viruses stick to the surface. So here on the right is the structure of the distal part of the neuraminidase, and here is sialic acid fitting into the pocket. So this, the substrate for neuraminidase is sialic acid. The crystal structure of this protein with sialic acid was solved. The structural biologist looked at it and said, let's design a small molecule that looks like sialic acid but isn't, which would stick in there with high affinity and inhibit it. So that's what they did. This is one of the first examples of structure-based drug design. So they made a small molecule that inhibits the neuraminidase. Actually, 2 companies made them, and they're both on the market today, zanamivir, Relenza, or oseltamivir, which is Tamiflu. 2 different molecules, 2 different companies, both of which mimic sialic acid. They fit in the pocket in the neuraminidase, and they block its activity. These things fit in, and the neuraminidase can't bind to sialic acid. So they're designed to mimic the natural ligand. The idea being, if it looks a lot like sialic acid, maybe the virus cannot change so that it's resistant, because then it wouldn't be able to recognize sialic acid, and it would be a dead virus, right? So that was the idea. Well, the upshot is that you do get a lot of resistance to Tamiflu, but not to zanamivir, not very much at all. And one of the reasons may be because these compounds are slightly different in the way they mimic sialic acid. So here on the left is our neuraminidase, the Y-shaped molecule on the virion, and the sialic acid binding. So the pocket is shown as a V-shaped pocket for illustrative purposes. Tamiflu is not quite V-shaped, right? So it's a little bit different from sialic acid. It deforms the pocket somewhat, whereas zanamivir more closely mimics sialic acid. And it's very easy to get mutations in the neuraminidase that block Tamiflu binding, but very difficult to get mutations for zanamivir resistance, perhaps because it's closer to sialic acid. And these are some of the changes, the amino acid changes that can happen in the neuraminidase to give you resistant viruses. So I took a few numbers from the 2007-2008 influenza season just to show you what happens. So this is pre-pandemic influenza. These are just seasonal viruses. 557 influenza A and B viruses isolated. 38 of them, 6%, were resistant to Tamiflu. And these were all H1N1 viruses, not H3N2, which were still sensitive to zanamivir. So all of these 38 which were resistant were still sensitive to the other drug. 429 A isolates looked at, 24% are resistant to amantadine. These were all H3N2 viruses. So the H3N2s which were circulating were mainly resistant to amantadine, the H1N1s to Tamiflu. In the next year, 100% resistance of the H1N1 seasonal strains to Tamiflu. Now the current pandemic strain is less. Yeah, I think I have the numbers here for you. So these are now antiviral resistance of samples since September of last year. So this is the line we want to look at. 2009 influenza A H1N1. 1,600 samples tested, and 1.2% or 52 are resistant to Tamiflu. So not a lot, but it's growing, and it will increase for sure. None resistant to zanamivir so far. They're all resistant to amantadine, the channel blocker, 99.8% resistant. And there's very little of the other strains of influenza circulating. The old seasonal flu, there's very little. The old H3N2, there's very little as well. So next season there will be more resistance, no doubt. I wanted to go over one other inhibitor with you, and this is an inhibitor of picornavirus uncoating, which was discovered in 1985. And I like the first sentence of this article here. This was done up in Rensselaer, New York. Any fans of Rensselaer here? No? The discovery of antiviral drugs known today can be attributed primarily to the tenacity of researchers and the serendipity of circumstances. This is a good summary of antiviral discovery back then. It's changed a lot now, and it's a lot more focused. But look, several years ago, a program was initiated to identify compounds which demonstrate antiviral activity against any of a number of RNA or DNA viruses. First compound to show activity was the diketone shown down here. So they had a big chemical collection. They were just screening for inhibitors. They found this one that worked against a rhinovirus. Following this discovery, several structural modifications were made to examine the following parameters. So they made all these derivatives of the original compound. The chemists went in and added side chains or rings and so forth. And they simply looked at the minimal inhibitory concentration in micrograms per mil. And you can see some of them were not active at all. Some of them were more active than others. And they eventually picked the best one and went forward with it. And this was the genesis of these Win compounds. I think I've told you about them before. These compounds bind into the capsid of picornaviruses in a hydrophobic pocket just below where the receptor binds. And here is the structure of one of these Win compounds binding in this pore. It fits tightly in this pore and prevents the virus from undergoing conformational changes that it needs to undergo to uncoat. So this is an uncoating inhibitor. Unfortunately, this never made it to be approved because resistance emerges very readily with the use of these compounds. It was tested for rhinovirus activity, but there's so much resistance, and the rhinovirus cold is of such short duration that it never made it to market. But these are used in the lab to study the process of uncoating. There are many new targets that are being explored. There's, for example, the hepatitis C virus protein shown at the top is a polyprotein. It's made as a precursor. It's then cleaved by 3 different kinds of proteases. There's a viral serine protease here. There's a viral metalloprotease here. And then these cleavages are done by a host signal protease, the host signal peptidase. Would that be a good target for developing an antiviral, you think? The host signal peptidase? No. Probably have really bad side effects because you need that to cleave signal sequences from proteins. And this one was shown very early on to be essential. If you deleted this protease from the viral genome, it wouldn't replicate. So this is now the target of antiviral drug discovery for hep C. On the bottom is another protease from a different virus. This is the herpes virus cytomegalovirus. This is a very unusual protease structurally. There doesn't seem to be anything like it in the cell. So the idea would be that if you could find a specific inhibitor, it might not have side effects. But again, you don't know until you put it into animals. There are clinical trials ongoing for the hep C drugs. I wanted to show you. You can find these announcements very easily online at various websites. And these are 2 companies starting a phase 2 trial, larger numbers of people answering different questions of a protease inhibitor. And also there are polymerase inhibitors for hep C being developed as well. And these are oral hep C drugs. There's also mRNA antivirals that are being tested. MicroRNAs are short 22 nucleotide RNAs that bind target sequences and cause them to be inhibited in a variety of ways. We've talked about translational inhibition mediated by microRNAs. And in this experiment, they took the hepatitis C virus RNA and designed specific microRNAs to match sequences in the genome. And these inhibit virus replication. So for example, here is an assay for virus replication. This is a northern blot type of assay. You can see here the viral RNA is replicating quite nicely. If you add a microRNA against GFP, it doesn't affect viral RNA levels. But if you add a virus-specific microRNA, you block virus replication. So the microRNA, again, binds to the viral genome and inhibits it. And these are being used or being considered for clinical trial as well because these can be delivered in a way that makes them quite stable. So microRNA targets for some viruses. HIV antiviral discovery is the most extensive of any. There are many targets for intervention of HIV replication, including attachment, reverse transcription, integration, transcription and post-transcriptional processing, and assembly. And we're going to go through a couple of these as well. The problem with AIDS is that you have years and years of virus replication. Remember, 10 to the 10th particles a day are produced. And we'll look at the numbers, but that's almost impossible to get around. The first HIV drug was AZT, which is a thymidine derivative. And the way it works is that it's phosphorylated. AZT is taken up by the cell. It's phosphorylated by 3 cellular kinases, so there's no restriction to virus-infected cells as there is for herpes simplex virus. And then the triphosphate drug is incorporated into DNA and causes chain termination because there's no hydroxyl here. There's an azido group. That's the Z and the AZT. So it's chain terminator. Initially discovered as an anti-tumor compound, actually, and then was checked for HIV inhibition, and it worked, and is a chain terminator. The reason it is somewhat specific is that it's not a great substrate for cellular polymerases. It's a little better for the reverse transcriptase. So it's somewhat selective, but there are many, many side effects of AZT because it does inhibit cellular polymerases. So unlike acyclovir, it has substantial side effects. The other problems, it's taken orally. It's got good bioavailability. It's absorbed rapidly, but the half-life is horrible, an hour. So you have to give doses 2 to 3 times a day. And this is problematic. It makes you feel lousy. It causes anemia. There are many side effects. And this kind of regimen, short half-life with multiple dosing, is a good way to select for virus mutants. And in fact, that happened immediately after the drug was initially licensed. Mutants resistant to the drug arose. These contain single amino acid changes at various places in the reverse transcriptase so that the drug the phosphorylated drug would not be incorporated into the DNA. So the altered enzymes don't bind the phosphorylated drugs. As a result, new analogs were developed, chemical modifications of various sorts or modifications of other nucleosides, and some of those are listed here. And that allowed double combination therapy to be used, the use of 2 antivirals at a time. Nevertheless, within a year, even using 2 drugs, double resistance arose. So this led to the development of non-nucleoside inhibitors of RT. So these are drugs or compounds that don't mimic nucleosides, but rather they fit into the reverse transcriptase and they block its activity. So here's the structure of reverse transcriptase. The polymerase active site is right here, and these non-nucleoside inhibitors bind a short distance from the active site. So they probably perturb the active site enough so that it doesn't work and inhibits the enzyme. And there are a couple of these that are being used at the moment. Now also, you get resistance to these, of course. Very rapidly, amino acid substitutions in the enzyme that prevent the drug from binding or modify the structure of the enzyme even when the drug is present. So you can't use these by themselves, and they're typically used in combination therapy. Now, when you have so many drugs that are available, you can then see if resistance to one drug does something with respect to resistance to another. And you want to know this because if you have a patient that becomes resistant to one drug, you don't want to give them another drug to which they're already resistant. So you need to know their resistance profiles. And some surprising results have emerged. And these are all examples of where resistance to one drug actually restores susceptibility to another. So here we have, for example, a change which confers resistance to this inhibitor. And then if you put them on a second drug, didanosine, and you get resistance to that, it restores susceptibility to the original drug. So this is the case for all of these compounds. The resistance to a second compound restores susceptibility. Yeah.
So does that mean that if you were putting someone on heart, that you would try to use drugs which have like this seesaw thing going on?
Well, in heart, there are only 6 combinations that are approved, and these are some of the considerations that are part of that. Yeah, you want to make sure that you're not going to get, if resistant to one doesn't give you resistance to all 3, but also some of these considerations come into play as well. So actually, you don't make up your own combinations. You have to use pre-approved ones, and those issues have come into play. In fact, nowadays, if you are diagnosed with AIDS, you have to have a resistance profile done. The best way is to sequence the genome in all the areas of known resistance and see what mutations you have, and then pick the treatment based on the sequence. And obviously, the more sensitive the sequencing, the better off you are, because there can be very small variants present which might not be detected unless you're doing very sensitive sequencing. There are a number of anti-HIV drugs that target the protease. Remember, the virus matures by assembly of a polyprotein that's processed by viral proteases, and the virus buds at the surface. One of these proteins is the protease, PR, the yellow circle here. It's incorporated into the particle and processes the polyprotein as the particle matures. And very early on, it was shown that this protease was essential, so it was used to develop antiviral drugs. A key discovery was that it would recognize small peptides. You didn't need a whole protein substrate to be recognized, but you could have a small peptide, and that the active site of the protease contained 7 amino acids of the substrate, so that was also useful. So initially, a number of inhibitors, protease inhibitors, that had already been investigated in hypertension studies provided the first leads for this work. These protease inhibitors actually inhibited the HIV protease. These are peptidomimics. They mimic the natural cleavage site. So these were the first drugs, and then subsequently mechanism-based screens and other structure-based designs were used to discover more. So here is the sequence of the protease substrate. These are the amino acids that comprise one cleavage site in one of the viral proteins, and this is the recognition sequence here. And then this inhibitor on the bottom mimics this protease cleavage site. You can see here, and then it's different on either side. So this binds very tightly to the protease. It's a peptidomimetic. It mimics the substrate and inhibits its activity. So now we use combination therapy, triple therapy, because the resistance, the probability of resistance is just the product of the individual resistances. So if you have two drugs with probabilities of resistance 1 in 10 to the 3, 1 in 10 to the 4, you need to have 10 to the 7 virions to get resistance, which is not a lot considering you get 10 to the 7th, 10 to the 10th particles made a day. And typically you target different mechanisms in this cocktail. And as we've said before, that way you can treat the infection as a chronic disease. And very recently, one pill was approved that has all 3 inhibitors in it, which makes it much easier than having to remember all the combinations on a daily basis. So here are the mathematics of drug resistance, which are worth going through. If you assume that 1 mutation is needed for drug resistance, which sometimes is true, but not always, and if the virus mutates 1 in every 10 to the 4 bases, which is once per genome per cycle, then in every 10,000 viruses, every base is substituted. So any pool of 10,000 viruses that you put out has a change. In each virus, there's a change at a different position in theory. So you make 10 to the 10th new viruses a day. So that means 10 to the 6th viruses are produced each day with resistance to 1 drug, 1 million. That's why 1 drug doesn't work very well. Now, if you take 2 drugs, 10 to the 8th viruses, and it's divided 10 to the 10th over 10 to the 8th, 100 viruses are made each day that are resistant to both drugs. So that's a little better, but eventually you will get resistance in about a year. And then if you do 3 drugs, the rate is 10 to the 12th. So you need fewer viruses, but eventually you will get there as well, depending on how much the growth is going on. Remember, replication is suppressed, so the number of 10 to the 10th particles a day is going to be brought down significantly by triple therapy, so it will take much, much longer to get resistance. My understanding is that you do get resistance, but they are mostly a consequence of people stopping their therapy for whatever reason, and the virus grows and the resistant mutants grow back. Some new antivirals have been developed recently. One of them is the integrase inhibitor that we have mentioned a couple of times in this course. Remember, the viral DNA integrates into the host DNA, and this integrase protein mediates the strand invasion by the viral DNA. The integrase inhibitors simply bind the integrase and prevent this joining, this nicking and joining reaction. And this is an example of one of the integrase inhibitors. You can see it's a small molecule that forms a variety of interactions with specific amino acids of the integrase. Now, of course, you can get resistance to these as well. And those have been well described in the literature. You can see here integrase inhibitor resistance mutations. There are 2 different integrase inhibitors shown at the bottom here, and all the different amino acid changes that you can get to give resistance. I thought I would show you this study, which is one way that you test an antiviral drug in a clinical trial. So this is a double-blinded, randomized, placebo-controlled trial, a dose-ranging study, different doses to find out which one is good. And they put 200, 400, and 600 milligrams of raltegravir and, of course, the placebo. And these are blinded, so nobody knows who's getting what. And then they measure the viral RNA, either the change in baseline or the proportion of patients with RNAs of different copies per mil, less than 400 or less than 50. So this is the gold standard, getting less than 50 copies per mil. And then the change in the CD4 cell count. So you can see these are the numbers for the placebos, and if you compare the drug treatment, just take the highest amount of drug, 600 milligrams, you can see that this is a big change, a negative change in the baseline of RNA compared to the placebo. Proportion of patients with less than 50 copies per mil, 66% compared to 13%. So you can see this is quite effective, slightly more effective than 400 milligrams per mil. 400 milligrams. This is an inhibitor of CCR5, which we talked about a long time ago. CCR5 is the co-receptor, remember, that is needed for HIV infection. It's a small molecule that binds in CCR5 and causes a change in the molecule such that the virus doesn't undergo high affinity binding. But of course, you get resistance to that as well. And finally, there are fusion inhibitors. These are actually peptides, 36 amino acid peptides, which interfere with the fusion reaction. So normally, when fusion occurs, here's the viral membrane and here's the host cell target. GP120, or the envelope protein, binds the receptor on the host cell. The fusion protein is exposed, inserts into the cell membrane, and then remember, there's a conformational change in this protein. It hairpins, draws the two membranes together so that they can fuse. We looked at movies of this a long time ago. These peptides, what they do is they bind these alpha helices that have extended here in the glycoprotein. So these small cylinders are the peptides, the antiviral peptides that are binding, and they lock it into this position so it can't undergo the hairpinning and the fusion. So these block fusion. These are very expensive drugs, and they have to be reconstituted just before injection. So you get them as a dry powder, you add buffer to them, and you inject them. And it's very expensive. And of course, we get resistance by amino acid changes along these alpha helices that simply prevent the binding of the peptide. And some of these are just examples of the kinds of mutations that you get. These have been well studied. Changes in GP41, which is the subunit of the glycoprotein to which these peptides bind. And these are all tracked at a website, the HIV-DB, where you can look at all the resistance mutations in real time. So this is the chart of all the available HIV antivirals at the moment. They're classified according to the mechanism. So you can see nucleoside RT inhibitors, non-nucleoside inhibitors, protease inhibitors. Then we have our fusion inhibitor we just talked about, the CCR5 inhibitor, integrase inhibitor, and then various combination products. So this is the tripla, which is the pill that contains 3 different drugs, and then other combinations can be given separately. What's interesting about this table is that you can see the very, very rapid approval rate for most of these drugs. It usually takes much, much longer to have drugs approved. So this is the time to approval where you submit your data to the FDA and they say it's okay. And this, of course, being an urgent situation, they're all fast-tracked and given fast-track type approval. So some of them are just a few months to approval, which is amazing. So you see we have a big arsenal, and that allows it to be effectively treated. Yes?
You might note that there's now quadrivalent guideline.
Is it, sorry?
There's a quadrivalent.
4 drugs, yeah?
Yeah, phase 3.
Okay. And what would be the resistance to 4 drugs? It'd be very high, but eventually you'd probably get there. But remember, this is why a lot of genomes on the planet, and no matter what you make, you're going to get resistance.